Hello, welcome to the Medchem Express MCE webinar series. This is David Lee. So first of all, I want to say good morning or good afternoon or good evening, depending where you are. And thank you very much for spending the time to join us today for this exciting webinar. So today, we are very fortunate to have Dr. Xin Lu from Notre Dame University joining us, and his webinar topic will be about cancer immunosuppression and how to conquer it. So first of all, I would like to give a brief introduction to Dr. Lu. So after finished his bachelor's study at Tsinghua University in China, Dr. Lu came to the US and pursued graduate study at Princeton University, Department of Molecular Biology, under the mentorship of Dr. Yibing Kang. And in Dr. Yibing Kang's lab, Dr. Lu's focus was on breast cancer mechanisms governing breast cancer metastasis. And he published many exciting papers that elucidating the both cellular and the molecular mechanisms that related to breast cancer bone metastasis. And after finishing his PhD at Princeton University, Dr. Lu joined another giant in the field, Dr. DePinio's group in MD Anderson. And in Dr. DePinio's group, Dr. Lu's work was mainly focused on the prostate cancer development. And also he published very important papers that elucidating the molecular mechanisms governing the prostate cancer development, and also shed important lights on the novel therapies, the novel therapeutic interventions on prostate cancer treatment. So after the postdoc training, Dr. Lu joined Notre Dame University, Department of Biological Science. And right now he has already been a tenured professor. So, and in Dr. Lu's lab, he's running several exciting projects that with the main focus on tumor and the crosstalk between cancer tumor cells and the tumor microenvironment, with a main focus on the immune cells in the tumor microenvironment. So his research has the goal of elucidating the crosstalk between cancer cells and the tumor microenvironment, and how the immune cells in the tumor environment govern the cancer immunosuppression metastasis. And by understanding these mechanisms, Dr. Lu's long-term goal is to be able to facilitate the development of effective novel therapies that will be able to enhance the efficacy of cancer immunotherapy. Well, with that being said, we are very glad to have Dr. Lu here. And Dr. Lu, so you have the stage now.
Great. Let me share my screen. Hold on. Let me do this. Can you see my screen?
Yes.
Okay, wonderful. Yeah, first of all, it's my extremely good pleasure and honor to present on this platform. And then thank you, David, for inviting me, and thank MCE team to support this talk. So yeah, so it's my great pleasure to introduce everybody about our research on cancer immunosuppression, and also the approach we have developed to conquer that. So without further ado, I will get started. I don't have a conflict interest for this talk. So in this war against cancer that we are all in, the priority is to understand who our friends and who our enemies are. And then that's the fundamental reason for us to study tumor microenvironment. And then, as we all know, a few years ago, the Nobel Prize was awarded to two pioneers in the cancer immune checkpoint therapy field, Dr. Jim Allison and Dr. Tadrugo Honzo, for their discoveries of the CTLA-4 and the PD-L1 checkpoints of the T cells, and also development of the therapies against them, eventually lead to a big breakthrough for the cancer immunotherapy field. And then this is a snapshot of the response rate of patients of different cancer types to the same PD-1, PD-L1 therapy. As you can see, like some tumors clearly have a pretty good response, closing to 50% or even higher. Or some other cancers, such as prostate cancer and pancreatic cancer, has a very low response rate, and sometimes it's single digit or even lower. The bigger question is what's the fundamental underlying mechanism for this resistance, and how can we apply additional therapies to sensitize such resistant tumors to immunotherapy. So the goal for us is to lift this flat tail of immunotherapy to a higher level. Right? For melanoma, this tail exists. It's about 10, 20, 30%. We want to lift it higher. For some other cancers, such as advanced prostate cancer, is still no tail yet. So we want to lift it up. Eventually, we want to bend this survival curve to benefit more patients. So as we all know, tumor is not just a bag of cancer cells. It's a very complex organ, and including not only cancer cells, but also immune cells, fibroblasts, and endothelial cells. And within the immune compartment, there are also different subsets. And then there are some, we can call them good cells, good immune cells, such as CD8 T cells, natural killer cells, dendritic cells that can do the cross presentation. But also there are some maybe bad immune cells that actually favor tumor growth. For example, regulatory T cells, myeloid-derived suppressor cells, or MDSCs, and a subset of macrophages, and also neutrophils, which often take the function as MDSCs. So in our lab, so basically we can think those good cells as our friends. We want to help them. We want to support them. And the bad cells, we want to think of them as enemies. We want to deplete them or inactivate them, or maybe redirect their differentiation so they become good tumor eliminating immune cells. So in our lab so far, we focus mainly on the 3 cell types: the cancer cell themselves, the CD8 T cells, and also the neutrophils, which, as I said, often behave as MDSCs. So yeah, so basically my lab has focused on the crosstalk between the cell types, and also the indirect or direct mechanism, how they regulate each other. And today I will highlight some of the work we've done, and to hopefully deliver some messages we'll summarize at the end. So first, let's look at how the neutrophils, or also called as the polymorphonuclear MDSCs, can suppress T cells. So we wrote a review, and also there are many other wonderful reviews as well, to summarize this process that, as we know so far. Clearly, during the tumor development, the solid tumors, they will have some cytokines and growth factors secreted from them that will engage the progenitor cells in the bone marrow to undergo this granular poiesis. And this granular poiesis will produce a lot of neutrophils, and some are mature, some are immature, and they will exit the bone marrow, go to the blood circulation, and then eventually go back to the tumor. And then once they're in the tumor, they can have a lot of functions, and some of those are associated with the immunosuppression, such as they can produce those immunosuppressive cytokines. They have enzymes that can deplete the essential nutrients to make T cells suffer. They can also have a lot of ROS and RNase based mechanisms to either directly kill, or maybe make the T cell become exhausted. They also have some unique phenomena such as mitosis to influence metastasis and other immune cells. So all these mechanisms can eventually lead to more tumor growth, and then the dysfunction of T cells. So a few years back, when I was at Ron DePino's lab, we made a few findings to elucidate this phenomenon where MDSCs, which actually majority of 95% of the neutrophils, those cells, that they can actually be targeted by some multi-kinase inhibitors, such as carboplatin, and also some chemokine receptor antagonists, such as this drug called ISX682, which is an anti-CSCR1/2 drug inhibitor. And basically those drugs, each of them can do something to those MDSC cells, make them either with very low frequency or reduce their activity. So when you do this together with anti-PD-1 and anti-CTLA-4 antibody treatment for this very aggressive metastatic prostate cancer, CRPC prostate cancer, you will have this synergistic effect to lead to the tumor eradication. So this is the main finding a few years back. And then among these therapies, in my opinion, I think the most attractive two are this cabozantinib or the SI682, which is the CRCR1/2 inhibitor together with immune checkpoint therapy for CRPC. So I will just give some examples about the subsequent clinical development of this kind of combinations. So first, this cabozantinib plus immune checkpoint therapy actually was very successful for renal cell carcinoma. You can see this was a study published in 2021 that showed nivolumab plus cabozantinib outperformed the sunitinib, the standard of care for RCC, and eventually gained FDA approval for metastatic RCC. For the prostate cancer, the metastatic CRPC, so there has been some reports already. For example, this phase 1b trial combining the anti-atezolizumab, which is the anti-PD-L1, together with cabozantinib, showed some promising anti-tumor activity and acceptable safety profile. So right now, there's actually a phase 3 trial for this combination. And then the interim analysis, as reported last year in August, showed the promise. There's definitely some promise. Whether this will eventually going to pan out to success or not, we'll see. But I think the promise is there. The other combination I mentioned is the combination of the CRCR1/2 inhibitor together with PD-1 or PD-L1 therapy. So this company, they already work on several clinical trials and then in different cancer types. And then another company actually published a paper recently in Nature to show that the CRCR2 inhibitor together with this AR inhibitor enzalutamide have showed a very low toxicity and lower neutrophil infiltration and some good response. So this study hasn't incorporated the immune checkpoint therapy yet, but I think that probably could be the next step to do for this team as well, for this company to try this combination. I think this combination could be quite promising. So in my own lab at Notre Dame, so we have done this through basic research, but also studying clinical samples. First of all, we want to know where are those neutrophils, the MDSCs in the tumors. So we study some human prostate cancer samples with those markers that can define those tumor-infiltrating neutrophils or MDSCs. And then we clearly saw that those cells, they are mainly present in the stroma, not in the epithelial region, as you can see here, compared to the stromal and epithelial regions. Also, the number of those cells in metastasis stromal is significantly higher than the primary tumor stroma. So basically, we think that these cells, they are present in the metastatic prostate cancer in the stromal area, and their main function is likely to inhibit the T cells in there. And then in another cancer type, in bladder cancer, which actually have more immune cell infiltration, and then we actually develop this dual signature marker. One is based on the tertiary lymphoid structure, the TLS, and then there also another one is the PMN MDSC signature. So basically, with this dual signature, you are able to stratify the patients that undergo the PD-L1 therapy into this very good response group and also the not so good response group. So actually, you can sacrifice those. You can stratify those patients into 4 groups. The one that have high TLS signature, low MDS signature, do the best. The ones that have a low TLS signature, high MDS signature, do the worst. So this could potentially help to stratify patients for future clinical trials for bladder cancer, maybe some other cancers as well. So for the mechanistic work, so I will, for the ones that are published a few years ago, I will just do one slide to show what we did. For example, in this study, our postdoc did a wonderful work to illustrate a new mechanism to explain how MDSCs suppress T cells. In this context, he found that the reactive nitrogen species that are derived from the nitric oxide and the reactive oxygen species in the MDSCs is able to cause this nitration of a tyrosine residue of this ILCK tyrosine kinase in the T cells. And this modification will make those ILCK critical kinase in the T cell receptor signaling pathway dysfunctional. Because of this, the T cell receptor signaling pathway is impaired and eventually lead to the T cell unable to respond to antigen stimulation. So this is, I think, a pretty good mechanism to explain how MDSCs can inhibit the nearby, the neighboring T cells. And we also came across an interesting model where we developed this penile cancer serendipitously. So this actually is a rare cancer. The penile cancer is a squamous cell carcinoma of the penis for males, and it's a rare cancer in the U.S., but it's actually more frequent in some other countries. So basically, we found that in this genetic model that allows SMAD4 and APC in the epithelial region of the penis, which is also actually responding to the androgen signaling. That's why we kind of stumble into this model, because we want to model prostate cancer, but later this mice actually develop a penile cancer. But the bottom line is they develop aggressive squamous cell carcinoma of the penis in the mice, and we look at their immune cells, except for the MDSCs, the neutrophils, which actually increase a lot. All the other immune cells decrease. Right? So this kind of really give us a hint. We can inhibit these cells, and then we look at for some efficacy. So in this story, we inhibit them with either the cabozantinib, as we did in the prostate cancer, because they have this high RTK PI3 kinase signaling in them. We also can inhibit them with a COX-2 inhibitor, cyclocoxib, because we found they have a super high level of COX-2 in them, which probably responsible for producing some prostaglandins. And then basically, by doing either of this alone, there's little effect. The ICB alone also have very little effect. But if you combine them together, you have a much better effect to control the disease. So so this work illustrates that this MDSC or neutrophil function exists not only in prostate cancer, but also in some other cancers as well, in the solid tumors. So basically, we summarize some of the findings we had, also some other scientists in the field made in the genitourinary malignancies, which include the prostate cancer, bladder, renal, and penile cancer, about this in this review. So if you're interested about know more about how the MDSC do in these cancer types, you can read this review. Okay. So now I'm gonna start to talk about a couple of stories in more depth. So first is this story about how the cancer cells can have some genetic changes that can influence the immune regulation in the prostate cancer. So this study started from notifying that the human prostate cancer genome is not flat. It actually have a lot of amplification and deletions. And then when I was a postdoc, I started to look for those amplicons and try to look for some potential oncogenes. So we screened about 200, 300 genes, and then we found this one gene called PEGOPUS2 or PEGO2. So this actually is located in this 1q21.3 amplicon. And this paper, we reported this PEGO2 gene as an oncogene because we have those xenograft model to confirm that. And at that time, we knew that PEGO2 had functions to have either go through Wnt-dependent or Wnt-independent pathways to regulate the gene expression. And PEGO2 actually has a domain, the pitch domain, that can bind to this H3K4 trimethylation marks, and then bind to some other transcription cofactors, eventually influence transcription. So that's what was known at that time. So my student Yi was very interested in whether this gene also has some potential function to regulate the immune system. So basically, we made a few transgenic mouse models. We have this conditional knockout mice of PEGO2, a gift from Dr. Xing Dai at UCI, and then we cross this PEGO2 conditional knockout mice together with this P10 and SMAD4 double knockout mouse that are driven by this probation Cre or NCAC 3.1 Cre-ER models. So we, in our lab and some other labs, we use this P10 SMAD4 double knockout to model metastatic prostate adenocarcinoma. It's a very good model. So we just cross in this PEGO2 loss and see what's the influence. You can see the PEGO2 loss was very good, very effective. And we clearly saw that the tumor grows slower. You can see like the 5 months, the 2 months, 3 months double knockout tumors are similar at size with the 5 months triple knockout tumors. So the triple knockout, which has the PEGO2 knockout, clearly grows slower, and also the mice live longer. And this is true for both this probation-based driver, also the NCAC 3.1 inducible Cre-based driver. And this is the inducible Cre driver. You can see like the tumor of the triple knockout have a lower tumor burden. And because we have this luciferase allele in the model, we can also assess the metastasis. Both the metastasis to the lymph nodes and to the lungs were significantly reduced when the PEGO2 is lost. So this confirmed that PEGO2 is indeed a prostate cancer oncogen. So we did a single cell RNA sequencing for the triple knockout, double knockout tumors, and then we look at the immune populations. You can see among actually the first glance indicates probably there's not too big changes, but if you look more carefully, you can see some T cells, for instance, CD T cells, CD4 T cells, they all increase, and Treg decrease. So we follow this line of information. We look at the genes that are altered in the CD T cells, and then the T cells in the triple knockout tumors enrich for those pathways that are important for T cell activation. We also look at some cytokines that are involved in T cell activation, and they also upregulated in CD T cells, but also they all upregulate some exhaustion markers as well, like a PD-1. So this suggests to me that the T cells in the PEGO2 lost tumors, they're more engaged. They're more like doing things. They still fail to eventually kill all the cancer cells, but they start to get engaged. So we look at we confirm some of these results with flow cytometry. For instance, you can see here that the CD T cells, CD4 T cells, they all increase a lot in the triple knockout tumors, and the Treg decrease. And we look at some cytokine markers for the activating T cells, the interferon gamma, TNF alpha, they also increase significantly in the T cells in the triple knockout tumors. So we engineered another cell line model of prostate cancer called RM9, which is transformed with RAS and MYC. We express ovalbumin, the oncogen in them, and the ovalbumin antigen in them. We also knock out PEGO2 in them. We compare the infiltration of the CD8 T cell that specifically recognizes the ovalbumin with the tetramer assay. You can see like there's a significant increase of the CD8 T cell that recognize this ovalbumin when the PEGO2 was knocked out in the cells. So again, suggesting that the PEGO2 loss in the cancer cells encourage more T cell infiltration and maybe their activity as well. So we have cell lines derived from these tumors, and we call them PS or PSP. PS is p-tau2, PSP is p-tau2 PEGO2. And then we confirm that indeed the PSP tumors have higher CD8 T cells. But more importantly, we show that if you re-express PEGO2 again in these PSP tumors, you actually can reduce the T cell again. So basically, this T cell phenotype is really dependent on the PEGO2 presence or absence. And then, and this is also true for the RM9 models as well. So since the CD8 T cells increase a lot in the PEGO2 lost tumors, we assess whether they are functionally important by depleting them with this anti-CD8 antibody. You can see like if there is depletion in the PS tumors, there's no change. But if you do this with the PSP tumors, you can actually abrogate some of the effect of the anti-tumor effect by the PEGO2 loss. The tumor become a big again, right? So this is the PSP models. We try this in the RM9 model, which actually give even more dramatic result. You can see like we have this PEGO2 knockout cells in that model, and then we put them into either the black 6 mice or the nude mice, which do not have T cells. The result is dramatically different, right? In the black mice, the PEGO2 loss lead to tumor regression, but in the nude mice, which don't have T cells, there's no effect of PEGO2 loss, really suggesting that the PEGO2 function actually is through this CD8 T cell regulation. So not only did we see like the T cell number increase after PEGO2 loss in the cancer cells, we also show that the activity of the killing activity of the CD8 T cells also is enhanced when the PEGO2 is lost by using this OT1 T cell and killing the ovalbumin expressing cancer cell assay. You can see like with more and more of the OT1 cells, the cancer cells are killed because they express ovalbumin, but this killing was more effective when the PEGO2 was knocked out in the PSP cell. This was also true in the RM9 model as well. So all this kind of phenotype suggesting that PEGO2 loss in the cancer cells really increase the CD8 T cell frequency, but also their cytotoxic killing activity as well. So next, we did a lot of work to figure out the mechanism. Because the time is limited, I won't dive into all the details. But basically, we first did the RNA sequencing for the epithelial cancer cells sorted from those double knockout or triple knockout tumors. And then we also, and based on this profiling, we highlight a few genes we validate. And then we also perform a lot of gain of function loss function experiments. And then we focus on the PEGO2, and then we eventually lead to this pathway that can explain the function. And this was also informed a lot with bioinformatics and some chromatin association assays. You can see like basically the story is like this: the PEGO2 will bind to the histone mark, the H3K4 dimethylation or trimethylation mark, and then also recruit some other factors, particularly the PP3, together with this acetyltransferase CBP. And basically this recruitment of PP3 will activate PP3. And this activation together with the PEGO2 will drive the transcription of SP1. SP1 is another transfactor which can lead to binding to the promoter region of KIT. And the KIT will drive the expression of the important immunosuppressive enzyme called IDO1. And IDO1 will convert tryptophan into a number of immunosuppressive metabolites and eventually lead to exhaustion of CD8 T cells. So based on this mechanism, we hypothesize that if we're able to suppress PEGO2, either genetically or pharmacologically, we should be able to synergize that together with immunotherapy. So to test that, so first we use these knockout mice, knockout cell lines, the RM9 cell lines, together with immune checkpoint therapy, which is the PD-1 and the CTLA-4 double antibody. You can see like the ICB alone has some effect. The knockout, of course, also have effect as we showed before, but the combination actually lead to complete 100% elimination of all the tumors in this model. And it's very dramatic result. So this is the ICB therapy. We also tried PEGO2 knockout together with the OT1 adoptive transfer therapy. And you can see the adoptive transfer itself has some effect, just like what the PEGO2 single knockout has, but the combination has much better effect. So that's the T cell based assay. We also actually tried the MDSC targeting assay with the CSCR1 inhibitor, the SS682 that I mentioned earlier. So we you can see like the single treatment have some effect, but the combination has the best effect. When you actually knock out both PEGO2 together with the reducing the number of infiltrating MDSCs, and this correlates with also better T cell profile as well. So these results really suggest that PEGO2 genetic knockout is effective to enhance immunotherapy. But what about pharmacological inhibition? So there was a publication a few years ago to have this virtual screening for PEGO2 inhibition, and then there was a chemical called JBC117 was reported in this paper that can inhibit the PEGO2. So we synthesized this JBC117 and also its analog in our university, and we test those in vivo. You can see like indeed this JBC117 or its analog is able to inhibit the PS tumors which have the PEGO2, but cannot inhibit the PSP tumors which do not have PEGO2. And also these proteins such as KIT and IDO1, they all reduced by the JBC117 or its analog in the PS cell lines, but not in the PSP. So suggesting these drugs actually are quite effective and selective. So we combine this JBC117 drug together with ICB in this RM9 model and saw a pretty good combinatorial effect and correlating with a good T cell profile as well. And this is true for the other PS model as well, but also in this CRPC model. So this actually is a very good model. The big cap cell line transform with a high MYC expression, and then you can actually castrate these tumors, and then they will develop CRPC after a few weeks. And then we treat these tumors with ICB. You can see there's no response whatsoever, right? There's no response at all. Just like in many CRPC patients, they don't have response. And then the GBC itself has some effect, but the combination have a much better effect. So really suggesting that the CRPC can also be treated with this combination. So finally, we did some analysis of the PEGO2 expression together with the CD8 T cell cytotoxic signature, and we see like the high PEGO2 correlate with the lower CD8 T cell signature, and also the number of CD8 T cells is lower when the PEGO2 is high. And we collect some, we analyze some published data to seek for more evidence for PEGO2 function in the immune context. So in this CRPC clinical trial, where they treat the patients with the ipilimumab, the anti-CTLA-4 antibody, there was some effect in this trial, but basically what we did was we analyzed their RNA sequencing result, and we saw that high PEGO2 is correlated with a worse prognosis in this clinical trial. That's prostate cancer. And we also utilize this tool to analyze other cancer types. You can see that in general, across many cancer types, the high PEGO2 is correlated with the worse prognosis for either anti-PD-1 or anti-CTLA-4 antibody treatments. So these results indicate that PEGO2 is indeed a promising target for enhancing immunotherapy. So we published this work last year, and then and my student made this cartoon to illustrate if we target PEGO2 with inhibitors, we're able to kind of really enhance immunotherapy and antagonize the cancer cells. So okay, so hopefully this story give you a hint about how the genetics, the genotype of the cancer cells can really influence the phenotype of the immune system. So and this is one direction my lab is pursuing, is to understand more how the genotype of the cancer cells can shape the phenotype of the immune system in the tumor, and utilize this knowledge, because those genetic changes in cancer cells, they are more likely to be targeted successfully compared to some non-genetic based changes. So now I'm going to switch gear to talk about another more recent study we have, which is to go back to the neutrophils to see what their functions are, how they survive in the cancer, and how they suppress T cells. So this work was done by our postdoc Yun Zhao. So basically, Yun has a few models of memory cancer models in the mice, the EO77 model, the PYMT model, and also a PYMT based cell line model. In all models, we saw there's a big increase of neutrophils in the blood, in the tumors, the primary tumors, but particularly in the lung metastasis. You can see there's a big increase of neutrophils. And then we did a T cell culture assay and confirmed that those metastasis isolated neutrophils are able to suppress the proliferation of both CD4 CD T cells, confirming they are indeed PMN-MDSCs. And then Yun did this single cell sequencing, comparing the transcription of the blood neutrophils as well as the tumor infiltrating neutrophils. These are from the lung metastasis, from the primary tumors. You can see, like, if you look at this T-SNE plot, the tumor infiltrating neutrophils and the blood neutrophils, they're actually located in completely different islands, so suggesting they have very different transcriptome activities, even though they share the same markers, that they're all like Lat6G positive or CD11B positive. So we analyzed their transcription, and we clearly saw like the tumor infiltrating neutrophils enrich for some pathways that are relevant to those immune functions. And then particularly, I want to highlight this NERV2 pathway, because this will come up again very soon. And we look at also the genes, and then we highlight this gene called ARCOD1. And because this gene actually is the most upregulated enzyme encoding gene in the tumor infiltrating neutrophils compared to the blood neutrophils. And they are here. You can see, like, the teens express ARCOD1 at a very high level, and then the blood neutrophils is actually do not express ARCOD1 at all. So what is ARCOD1? ARCOD1 is aconitase decarboxylase 1. It actually was an enzyme recently discovered in the context of macrophages. You can see basically what ARCOD1 can be induced by pathogen infections, toll-like receptor ligands, and also some cytokines in macrophages. And the main function of ARCOD1, actually ARCOD1 enzyme, the function is to convert cis aconitate into itaconate in the mitochondria. And then itaconate in the mitochondria, the main function is have this anti-inflammatory activity in the macrophages. And this play important roles in immunity and inflammation. And then the function of ARCOD1 or itaconate in neutrophils was poorly understood. That's why we set out to study it. So first, you confirm that in both the primary tumors and the metastasis models, the neutrophils is the one that expresses ARCOD1 the highest, and then followed by the macrophages. And then he also sorted the different immune cells, including the neutrophils, macrophages, but also dendritic cells, T cell, B cell, etc. And then measure the ARCOD1 level. You can see neutrophils indeed express the ARCOD1 at the highest levels. And we extend the human breast cancer tissues, also confirm that ARCOD1 is expressed by the CD15 positive neutrophils in the breast cancer as well. So because there are all these wonderful publications of single cells of different cancer types. So we reanalyze this data from this group, and then which is the human metastatic breast cancer data set, and we found that there's one population in this immune cells that express ACOD1, and then we look more carefully, and this population are two cell types, the neutrophils and also the macrophages, and then most of the other cells do not express ACOD1. So, and also we compare the level of ACOD1 in the blood, also the the metastasis. You can see like just like what we saw in the mouse models, the metastasis in different organs, they express ACOD1 in the neutrophils at a higher level compared to the neutrophils in the blood. So this confirms that ACOD1 is indeed positive in metastasis infiltrating neutrophils. The next question is, do they have any functions? So we perform loss of function experiments. For the loss function, we use the whole body knockout ACOD1. That's where we started. We first use this ACOD1 available from Jackson Lab. We use these mice to confirm that the metastasis, the lung metastasis from the EO771 cell line indeed was reduced when the ACOD1 was lost in the host, and the mice lived longer. Next, we inject this EO771 based lung metastasis assay with this itaconate derivative called DMI. So when you inject with DMI, you will increase the lung metastasis signals, and the mice lived shorter. So because this is a whole body knockout and then whole body DMI treatment, we want to do this in more neutrophil specific manner. So first thing we did was we do this neutrophil adoptive transfer. So basically, we isolate the bone marrow cells from wild type or the knockout mice, and we isolate we treat them with the bone marrow with conditioned media from the cancer cells, and then we isolate the neutrophils from them. These are the bone marrow derived neutrophils, and then we transfer them into the black sick mice that are injected with the EO771 cell line, and then we isolate their metastasis signals and the survival of the mice. You can see that transfer of both the wild type and ACOD1 knockout neutrophils are able to increase the metastasis, actually, both cases, presumably because those neutrophils, they behave as MDSCs. They suppress T cell, they can promote metastasis. But more importantly, we saw that the neutrophils of the wild type background increase even more compared to the knockout background. So you can see this followed with the survival as well. And then, so that's the adoptive transfer results. At least Western just confirmed that indeed, if we culture the bone marrow cells with the conditioned media, we're able to induce very high ACOD1 in the wild type neutrophil, but not in the ACOD1 knockout neutrophils. All right, so this actually is another interesting model where we cross the ACOD1 knockout mice into the MMTV polymorphism T spontaneous breast cancer model. You can see the metastasis also reduced a lot. So this model was used typically as the kind of the gold standard for metastatic breast cancer. So we confirm this ACOD1 loss also works in this model. And later we also start to knock across this inducible Cre-Lox ACOD1 allele with the LICE-M Cre or the MRP8 Cre, which can delete ACOD1 in neutrophils. You can see that indeed in this neutrophil specific ACOD1 knockout, we also saw elongation of the survival and reduce of the metastasis. So phenotypically, ACOD1 in the neutrophil indeed promote cancer. What is the mechanism? Because the time is limited, I won't dive too deep. But basically, we look at both the upstream and the downstream of ACOD1. For the upstream, we confirm that GM-CSF is a growth factor secreted by cancer cells, maybe some other cells too from the tumor that bind to the that activate the JAK-STAT pathway, particularly the STAT5 pathway, and turning on this transcription factor CBP beta, and CBP beta will bind to the promoter region of ACOD1 and drive ACOD1 expression. And you can see like the ACOD1 and CBP beta are located in the same group of cells, the tumor infiltrating neutrophils in our single cell sequencing. And if you knock down CBP beta with sgRNA, you will reduce the level of ACOD1. Okay, so that's the upstream. That's how the ACOD1 is turned on in the neutrophils. What about downstream? Downstream, we look at the phenotype of neutrophils when they lost ACOD1. We saw that first of all, their number is reduced. The neutrophil number is reduced when the ACOD1 is lost. Then we look at why that's the case. And turns out the main outcome of ACOD1 loss in neutrophils is because the increased fructosis. We have several new fructosis markers we detected in vitro and in vivo. We saw they are turned on by alcohol-1 loss. And then for this, I just show some data. You can see like in this in vivo data, where we have this alcohol-1 knockout that reduce lung metastasis. But if we treat mice with FER1, which is a fructosis inhibitor, you can rescue this phenotype. Basically, the tumor, the metastasis grow fast again, suggesting that basically turning off fructosis is actually the key outcome of key mechanism for alcohol-1 loss. Because once you rescue that, you lose this phenotype. You can see like what's the mechanism for this fructosis? We go back to literature. We found some papers that itaconate is able to inhibit KEAP1, which inhibit nerve2. So and then when itaconate is high, then nerve2 is released, and nerve2 can have this antioxidative gene transcription, and then we eventually can resist fructosis. So we confirm this pathway indeed takes place in neutrophils, and that's the mechanism how the alcohol-1 can support neutrophils escape from the fructosis. You can see when alcohol-1 is lost, nerve2 is low, and then also if you use some of the inhibitors, you can confirm this phenomenon. So the mechanism is there. What's the clinical implication of these findings? So we use these alcohol-1 knockout mice together, either alone or together with immune checkpoint therapy. And then in this EU771 lung metastasis model, you can see like each alone has some effect, but a combination, the alcohol-1 loss together with ICB, have a dramatic elongation of the survival of the mice. This is associated with lower neutrophils, higher CD8 T cells, higher interferon gamma, granzyme B, and the higher helper T cells, but lower Tregs. Basically, all the immune profiles favors tumor elimination when the alcohol-1 loss together with ICB. And we confirm this also with this MRP8 neutrophil-specific alcohol-1 knockout. And when we inhibit fructosis with FER1 in this model, you can see the survival benefit is weakened, right? So really suggesting the fructosis is a major mechanism to explain the phenomenon. So in summary, in this study, we confirm that tumor-infiltrating neutrophils can escape fructosis through activating the alcohol-1 dependent immune metabolism switch. And then alcohol-1 is likely a tumor-specific neutrophil target to offset this immune suppression and improve immunotherapy. So this paper highlights our findings. I think this alcohol-1 enzyme is indeed a very promising target because first of all, it's an enzyme, and secondly, it's turned on really only in the tumor infiltrating or metastasis infiltrating neutrophils, but not in the neutrophils in the blood, in the bone marrow, or other lymphoid organs. So it's a very cancer specific. So basically, there was a paper a couple of years ago that published to elude that a metabolite called citraconate is able to inhibit alcohol-1. So citraconate may or may not become a drug because it's actually a metabolite. So, but right now I'm working with our collaborators here to develop actually some inhibitors for alcohol-1 in our lab. So in the last couple of minutes, I want to highlight our recent findings just accepted in cancer research. Basically, this was done by our student Sean Murphy. So Sean was interested in ketogenic diet, and especially its influence in this tumor immune crosstalk. So what's the influence of this special kind of diet in the tumor immune microenvironment? So in this study, we utilize this model we developed in our lab. So basically, we have this PTSMAD4 PV3 based cell line. Well, they can grow in black mice, and they actually respond to the single or double knockout or double immune checkpoint therapy pretty well. But then after they go undergo this in vivo selection process, we develop this PBS ICB resistant cell line, which no longer respond to ICB therapy. So this is the model we use for this study. So first of all, we profile those cell lines. We saw some up and down regulated genes. So we're still going after some of these genes to try to find what causes resistance. But using this ICB model, we start to use it. We started for this study. You can see so what is a ketogenic diet? So compared to the American diet, which has very high carbohydrate, the ketogenic diet has a very high fats, a very low carbohydrate, and a little bit of protein. So basically, that's the composition of this ketogenic diet. If you look at the PubMed, actually, there's a definitely rising interest in ketogenic diet in different contexts. So basically, once a person eat ketogenic diet, those fatty acids will be converted into ketone bodies in the liver, and particularly this ketone body called beta hydroxybutyrate, and the BHB and some other ketone bodies will be transported in the blood and go to different organs to serve as energy. And basically, ketogenic diet has been used to treat childhood epilepsy. It was shown to elongate the survival of mice, so it could have some health benefits. And also, some people actually use ketogenic diet to lose weight. And the ketogenic diet may employ this metabolic difference between cancer cells and normal cells to target cancer because of this Warburg effect in the cancer cells. But there's not too many studies to discuss the ketogenic diet in the immune context yet. So basically, in our animal experiments, we apply this standard diet, similar to the American diet, and also the keto diet, similar to this, and then in the mice. So first of all, you can see like the ketogenic diet we use have 90% fat, 10% protein, and no carbohydrate. So we test this keto diet in mice, and clearly the mice don't like it, and they lose weight. So and then Sean actually come up with this idea to cycle the keto diet and then the standard diet. Basically, 5 days on the keto diet and 2 days on the standard diet, and doing this in cycles. And this is called a cyclic keto diet, CKD. So when he did this in these models, you can see clearly, first of all, in the ketogenic diet cycle, there's a big increase of beta hydroxybutyrate in the blood from very low into like 3 millimolar. So this is the level that consistent with the level of BHB in humans when we eat a ketogenic diet. And then, but once the mice are in the standard diet cycle, the 2 days in the standard diet, the level drop a lot. So this increase clearly is reversible. But impressively, with this ketogenic diet, the tumor really cannot grow very well. You can see like just with this diet alone, the tumor already grow poorly. So we look at the mechanism. So there was a previous science paper indicating that the beta hydroxybutyrate actually is an endogenous histone deacetylase inhibitor, the HDAC inhibitor in vivo. So we confirmed that indeed, both in the cell lines we treated with BHB, but also the tumors isolated from those mice, there's an increase of histone acetylation and also acetylation of other proteins as well. So, but importantly, this histone acetylation can drive the MCE class 1 expression. We look at both MCE class 1 genes in the mouse cells, and there's a definite increase of MCE class 1 in the tumor cells treated with the cyclic keto diet. So this can potentially increase the antigen presentation. And we saw like this effect, this CKD effect, is really dependent on the adaptive immune system, because we use both the BATF3 knockout mice, which do not have the dendritic cells that can do cross presentation, and also we use the regular mice, which do not have T cell and B cells. In both cases, the CKD effect was gone, right? So suggesting that this tumor antigen presentation and the adaptive immune system is crucial for this phenomenon. And then we look at the ketone body, the beta hydroxybutyrate, and using this BDH1 knockout mice. So basically, as I mentioned, in the liver, the fatty acid will be converted into acetyl and eventually become a beta hydroxybutyrate with this enzyme. So if you knock out this enzyme, you no longer have this beta hydroxybutyrate in the system. You can see like we confirmed that we use the BDH1 knockout mice, the level of BHB dramatically reduced, even they undergo this CKD. So we confirm that when the knockout the BDH1, the tumor reduction benefit by the CKD is also gone, suggesting that the BHB is really the key factor there. So because of this, we actually come up with another diet solution. We just mix this precursor of BHB into the regular diet, into the standard diet. We call it a BD, like a BHB or the mixed diet, BD. And then in this BD diet, which is actually easier to give, right? You just mix this with the standard diet and give to mice. You don't have to do this cyclic ketogenic diet. And in both cases, we did a single treatment and also together with ICB treatment. The ICB alone has very little effect, as I showed earlier in this model. And then the CKD and the BD each have some effect. Actually, the BD have even better effect than the CKD, but the combination have the better effects. You can see like it's particularly with this BD plus ACB combination, about 25% of mice actually were cured by this combination. So what's the effect of this combination in the immune system? So we did a single cell sequencing of the standard diet, the BD, the standard diet plus ICB, and the BD plus ICB. So you can see like in each condition there's changes, but the most dramatic change is coming from this BD plus ICB combination. You can see this combination lead to a very high CD T cells and the increase of this monocyte population, which can potentially give rise to dendritic cells and the M1 macrophages, as we did with this, the lineage tracing analysis of the single cell data, and also increase of this dendritic cells, increase of M1 macrophages, and decrease of this M2 macrophages, and decrease of some neutrophils as well. So basically, all this combination really favor this high T cells and low immune suppression profile of the immune cells in tumors. And then we look at T cell particularly, we found that we use this cell chat analysis to find what actually potentially drive this increase of T cells. And it turns out this chemokine receptor called CXCR3 is a big attraction for CD T cells. So we confirm this using the anti-CD CXCR3 antibody. You can see like if we block the CXCR3 antibody, we can clearly abrogate the effect, all the effect coming from this BD plus ICB combination. So this is the combination of BD plus the immune checkpoint therapy. What about other immunotherapies? So in this case, we turn back to this mid-cap model. And previous, there was a couple of papers to show that in this model, the T cell number is very low. However, there's a population of B cells. They are immunosuppressive B cells, and they can actually promote CRPC in this model. And then we have this anti-CD20 antibody that can deplete this immunosuppressive B cell population, and we combine that together with the BD. You can see like the BD alone has some effect. The anti-CD20 antibody also has some effect, but the combination have really much better effect. So I think this suggests that this the BHB administered diet, the supplement diet is able to help not only in the ICB therapy but the other potential immunotherapy as well. So in summary, in this study, we found that either you can do the HDAC inhibitor or the cyclic keto diet, or this combination, this supplement diet, you are able to elicit this intrinsic mechanism to suppress HDAC and turn on IMC class 1, but also extrinsic effect to reprogram the immune system to make them anti-tumor. And then there's actually a recent paper published to show that BHB actually is favored by CD T cells over glucose to produce acetyl-CoA and then for metabolism and cytokine production. So really suggesting that if we increase the BHB level in the tumor microenvironment, we can really promote the anti-tumor T cell immunity. So, okay, in summary, I hope my talk can deliver this message that we need to look at multiple players in the tumor microenvironment. We need to look at the monster cells, the cancer cells themselves, because their genotype can dictate the immune suppression. For instance, in the context of PEGO2, we target them, we can actually release more T cells. We also have a recent paper to show that particular HSP90 isoform called HSP90 beta is also a good target. And we also look at the T cells, of course. For example, we can have a better immunotherapy for T cells. We can have a different kind of engineered T cells, but also we can actually help them with some metabolites, for example, the beta hydroxybutyrate. But also we need to look at those saboteurs, because they sabotage this effort of T cells. Those neutrophils, we have to target them with either some kinase inhibitors or these chemokine receptor inhibitors, but also some emerging targets such as ACOD1. Some other players that can influence the host, such as the diet, the tertiary lymphoid structures, the nervous system, the microbiome, and many other factors also need to be investigated to eventually can enhance the immunotherapy and benefit more patients. So with that, I want to thank my lab members. particularly Yun and Yini and Shang, they did a wonderful work as I alluded, but many other lab members as well, and also my collaborators at Notre Dame and outside Notre Dame, and our funding resource. And my lab welcomes applications of PhD postdoc, postdoc fellows and PhD students. So if you're interested, please contact me. Okay. So lastly, I will leave this with these slides to introduce our university. Okay. Thank you very much for your attention.
Thank you, Dr. Lu, and thank all the audience for the attention. It's a great talk, Dr. Lu. So I think you should be able to see the questions in the Q&A.
Yes.
Yeah, so there are, because the time is limited, so we would focus on the questions that are kind of more related to today's topic. Start with the questions that are related to today's topic. So there are questions, for example, okay, so with ACOT1, so an audience asked, will ACOT1 signaling effects on the T cell exhaustion?
Yes, a great question. So ACOT1 signaling, because as I showed, ACOT1 is expressed pretty uniquely in the myeloid cells, particularly neutrophils and the macrophages. So they probably cannot directly, affect T cell exhaustion because it's not expressed in T cells. However, ACOT1 expression in the neutrophils is able to produce itaconate. So in our study, we show that this can help the neutrophil to survive in the microenvironment because they can no longer undergo apoptosis. And those neutrophils, they are MDSC cells. They are able to suppress T cells, lead to their exhaustion. So you can see that indirectly, ACOT1 signaling in the neutrophils is able to induce T cells exhaustion. Whether directly there's an effect, I'm not aware of. Thank you.
Sure. And another question is, do we have good understanding on how those neutrophils are recruited into tumor microenvironment? And can this recruitment or the infiltration process be targeted for therapy?
Right. So yeah, great question. So the neutrophil recruitment is definitely a hot topic. And then, so far I think the major understanding is that the regular signalings like CXCR2 particularly, sometimes CXCR4 as well, is also responsible for bringing neutrophils in the tumor as well. In most cases, if you target the CXCR2 with genetic or pharmacological approach, you can dramatically reduce the neutrophil number in the tumors. So I think the initial entry into the tumor is probably not too different than what neutrophils typically do. The question is, once they are stepped into the tumor, they will be emerged into this tumor, cytokine environment that will totally transform them and make them no longer look at the neutrophil. They are MDSCs, they live longer, and they have this reprogrammed metabolism. So the initial recruitment is probably not too different than the homeostasis. It's just exactly the version of that. But the thing is, like, after they get into the tumor, then things become really bizarre, and that's where the targeting can be really meaningful, right?
Okay, so there are more questions I think we can answer on time, but we may extend the webinar for a couple more minutes. Is that okay with you, Dr. Lu?
Oh yeah, sure, yeah.
Okay, so I think there are a couple of questions related to the role of neutrophils and also the ferroptosis. So one question is that so basically the ferroptosis, there's a recent paper that shown that ferroptosis can actually cause the immunosuppression in the tumor microenvironment. And in your talk, I think the ACOT1 loss would lead to increase of ferroptosis that actually are beneficial to the immune responses. So in this case, I think the question is, did you check the ACOT1 expression in different immune cells? And then basically would understand the role of whether whether the ACOT1 would regulate ferroptosis differently, or the ACOT1 regulation of ferroptosis has different outcome to the immunoactivation or suppression.
Right. Yeah, I mean, thank you for the question. Actually, I'm aware of this study. Was I think Nature paper, and talking about how the MDSCs can utilize ferroptosis as a mechanism to induce suppression. So actually, we discussed this in our paper as well. So I think this this study and our study reflect the two sides of the same coin, right? So basically, clearly, the neutrophils are present in the tumor. They're very highly abundant, and then there's no way they all can undergo apoptosis, right? They must have ways to invade from apoptosis. They survive there. So and our study point to that, how they do that. Like ACOD1 upregulation in the neutrophils help them to largely escape from this apoptosis, so they can still remain viable. And they have a PD-L1, they have arginase, they have many other weapons. They can suppress T cells. But those are causing immunosuppression. So but on the other hand, some neutrophil will die. And then when they die, one mechanism is apoptosis, and then those apoptosis brought this peroxidized lipids are also able to suppress T cells. So this kind of like, you see, like once the neutrophils in the tumor, it's almost like their whole life is dedicated to suppressing T cells. When they're alive, they have mechanisms to suppress T cells. When they're dead, their dead bodies also can suppress T cells, right? So basically, I mean, I think that's why I'm saying like our story and then the paper you alluded to actually reflect the two sides of the same coin. And I think together, if you look at them together, you put them together, this can really tell the whole story. And then whether targeting ACOD1 can cause complications or not, I don't know, right? We have to wait to see. In the genetic models, in the mice, they seem to be pretty good. I mean, the result is pretty good. But in the humans, I don't know. I mean, other cancer types, I don't know either. So it's really something we have to wait and see. Yeah.
Okay. So just extend a little bit from that. So one audience pointed out that the neutrophil has also context-dependent functions, tumor suppression or tumor promotion. So do you have any comment on that?
Yeah. So yeah, great question. Yeah. So yeah, neutrophil definitely have this dual phase. They can, under certain conditions, they can become kill tumor cells or as well. So in our models so far, we actually test this quite often, like whether the neutrophils will isolate from the tumors, primary tumor, metastasis, prostate cancer, breast cancer, other cancers. Can they have this tumor killing activity? And then usually we don't. Most time we don't see that. It's most likely they have these T cell suppressing activities. So that's why we kind of follow this line in most of our research. But I know there are papers, in the very high profile journals saying that if you treat them properly, especially with some combination therapy, you can reprogram them to become tumor killing. And then in our lab right now, we also start to engineer those neutrophils, also try to push that direction. So basically, instead of completely deplete them, we want to utilize them because they are abundant. So we can utilize them to turn their back to kill cancer cells. And then we are also kind of on that path. So yeah, it's a great question. And then both functions definitely coexist. And then in some contexts, one takes the upper hand. In other contexts, the other one takes the upper hand. And our hope is to encourage that tumor killing activity and suppress the T cell suppressing activity.
Okay, great answer. Thank you. So let's maybe focus the rest of the time on other stories in the talk. So there's one question asking related to PEGO2 story. So you talk about the PEGO2, which is mainly expressed in the tumor cells. The question is PEGO2 also expressed in tumor infiltrated lymphocytes and other immune cells, and specifically whether is it expressed in CD8 exhausted cells?
Yeah, great question. So yeah, I mean, we definitely look at that in the single cell sequencing data, our data, and other people's data, and then I think the result is still uncertain. So because you can definitely see some small populations in like T cells and some myeloid cells express PEGO2, but not like entirely. You never see like entire like certain immune population express PEGO2 very high. So because PEGO2 has a Wnt associated mechanisms, PEGO2 is actually in the Drosophila. That's how PEGO2 was initially discovered in the fly. In the genetic screen, they look for this this Wnt phenotype, and they found PEGO, and then it's definitely essential for the Drosophila Wnt signaling. And then you can imagine in the immune system, when the Wnt signaling is essential, it actually play a role during maybe certain differentiation or exhaustion stages, then maybe that's where PEGO2 can have a function, right? So we don't know for sure, because actually that's something my lab is trying to study right now, is to see, besides cancer cells, what are other roles of PEGO2 in the immune system. This is important because if PEGO2 has roles to positively regulate the immune cells, then targeting PEGO2 can be risky. But if it has a negative role to regulate the immune cells, then maybe targeting PEGO2 has a dual kind of this benefits that even not only inhibit cancer cell but also immune cells. That's something we want to find out very soon, right?
Okay, great. So last question about the keto diet story. So the question is, did you see any inverted correlation of keto diet with cancer progression in patients? I think the audience is also interested in knowing the clinical application of keto diet with cancer treatment.
Yeah. So yeah, I mean, thank you for the question. The ketogenic diet is first of all, it's not for everybody, right? So it does have it doesn't taste good to many people, and then for some patients that are already very weak, it's very hard to utilize this approach. So in the preclinical models, if you look at the literature, there are some papers that are suggesting the keto diet can reduce tumor, and there's some papers showing no effect. There's some papers showing opposite effect. And then there are some actually this meta-analysis papers that summarize like hundreds of those papers and eventually conclude that majority of the papers suggesting ketogenic diet have some anti-tumor effect. It's just the scale. Some are very dramatic, some are a little bit. So majority support that. So I think this probably has something to do with the the tumor models being used, right? So if the models has this Warburg-like effect, maybe keto diet will be more anti-tumor. If the model has have very like flexible metabolism, then they don't mind utilizing keto bodies, then maybe there's no effect. And then also whether it's have the immune system or not, that's probably also important to interpret those results. So for clinical data, I don't think there's like a very good results or conclusive result about whether there's a correlation or not between taking keto diet versus the outcome. It's just very difficult to do those studies. So, but if you look at the the clinical trial website, there are ongoing clinical trials. Many of those people are trying this cyclic keto diet, and some trying supplement as well, and trying to understand more because this is important. And then I think the most highlight of our study is that this supplement, if you can do supplement instead of using this whole ketogenic diet approach, it will be more easy to reach patient compliance and probably more likely to work. But still, it's gonna only work probably in the context of a combination with some therapy, not alone. Yeah.
Great. Well,
so I did see there's a question about this
yes.
radionuclide therapy. I'm sorry, I don't know much about it, so I cannot answer that. Sorry about that.
Okay. Well, thank you very much, Dr. Lu. Once again, thank you very much for this great talk and for this great discussion.
Invitation. I really appreciate it. Thank you so much for the invitation. Yeah.
Thank you. Yeah,
thank you, everybody. Have a great day.
Yeah, we thank all the audience. We are very grateful for your participation, and there will be more exciting webinar series in the coming months. I think at the end of April, there will be an exciting webinar focusing on the role of organoid and the organoid in both the mechanistic studies and also the therapeutic interventions. So please stay tuned for that talk. Again, with MCE, our goal is to not only just provide good products and good service, we also wanted to create a platform to facilitate scientific communication and facilitate scientific discovery. So this webinar is very important. So we really appreciate your continuous participation to this event. And if you have any questions or any suggestions, please contact us. Thank you very much.
Yeah, and also this will be videotaped and shared later, right? On the website, right?
Yeah. Yes.
Yeah. Thank you, everybody. Yeah, have a great day.
Thank you, everybody.
Thank you, David. Thank you for the support.
Thank you, Dr. Lu.
Take care. Bye-bye.
You too. Have a good day. Thank you, everyone.